Next speaker is July Wong. He's an informatics architect at Informatics Institute. He's basically our go-to guy for any question regarding U-BRITE and the infrastructure. He's also a fantastic Python programmer. So he'll be talking about our U-BRITE infrastructure. And the title of the talk is U-BRITE, a biocomputing infrastructure to accelerate data-intensive translational science. July?
Thank you, Malay. Appreciate the nice introduction. So good morning, everyone. Hope you're having a good ATTIS so far. So I'm definitely pleased this morning to get a chance to talk to you a little bit about U-BRITE, which is what we call our project to develop IT infrastructure in support of people doing data-intensive translational research. In particular for today, I'm just going to talk about what U-BRITE is and maybe show some examples of how it might help teams carry out team science. So I've just got a couple slides here. So U-BRITE is an acronym, and it actually stands for UAB, Biomedical Research Information Technology and Enhancement, which is a bit of a mouthful, so we all just call it U-BRITE. And it's definitely infrastructure about at least these three main things bulleted here. So one thing we're definitely trying to do is assemble clinical and bioinformatics tools that people think are important and make sure they're installed and work well on UAB's HPC infrastructure. So U-BRITE's definitely about getting the right tools in front of researchers in the right place. It's also about trying to help researchers with their data management and analysis pain points. So one of the big things we're trying to do with U-BRITE is experiment with and implement a design pattern called Commons. If you're not excuse me, familiar with that design pattern, I did put a quick definition from one of the first papers to describe this design, and that's Robert Grossman's paper from 2016. And it defines a commons as an infrastructure that co-locates data storage and compute with common analysis tools. So it's a seemingly simple idea that I think we can all get a lot of mileage out of. So you'll often hear us refer to U-BRITE as a translational research commons environment. And finally, we try to use U-BRITE to help facilitate team science, especially to the extent that technology can help people work together, excuse me, better as a team. So if you look at the overall architecture of U-BRITE, there's three main abstractions that we have implemented. So there's the idea of a data portal, there's an analysis gateway, and there's an expo gallery. And if you think about maybe a prospective new user coming to U-BRITE, how they might engage it, one of the places they might typically start is the data portal. A lot of users and teams might come to us with problems like siloed data or siloed code, or just generally ideas that they want to share with their team, get feedback, iterate on faster, but they're just being blocked for various reasons, right? So one of the things we try to do is encourage people to start to engage the data portal, right? So they might begin by putting their source code instead of maybe at their desktop or notebook computer somewhere, to actually put it in a source code repo and immediately start using the well-documented and well-understood workflows within the Git ecosystem to start sharing code both within their team and with other people. Similarly, if they've got data siloed off somewhere, to start to put it in the omics data repo, or maybe if you don't have your own data, you can query our many locally cached public data that we try to put there. And so that's, just gives you some ideas about the data portal. But once folks do that, then that implements kind of the first part of the commons, right? So you've put your data side by side with the biggest compute and storage here at UAB, which is the Chiha supercomputer that's depicted on the top right. And then you can start thinking about some of the stuff I personally find pretty cool, which is, well, what's the right analysis environment? What's the right abstraction? And so if folks are interested in a web notebook style UI, they might look at Jupyter Notebook. If they're interested in doing more of an interactive research tool, then they might look at Streamlit or RShiny. But regardless of what a team ends up choosing, what we try to do with U-BRITE is is assemble these tools, right? And so that includes tool building frameworks like Streamlit and Jupyter, but also just libraries of data science and AI and machine learning and visualization libraries, and try to make sure they run well within the commons. So before I leave this slide, right? So U-BRITE is about how this commons idea can help people achieve that fast R&D process that I think we all want. And part of how we do that, of course, is trying to remove obvious traps like those code and data silos, and maybe some of the obvious ceilings to scalability that we see teams run into, and try to provide a path to something that will avoid those. So for the rest of my 10 minutes, I'm actually going to do just a live demo. So we've got a what we call a U-BRITE technology demo, and that's located at demo.ubrite.org. And it's a live running web application that folks are welcome to come check out and play around with it. I wanted to show it today because it's a little bit easier to show some of this than it is to talk about it, especially some of the possibilities within the U-BRITE Commons. And so if you scroll down to the overview diagram in the demo, we try to highlight a few of the key interactions that we often observe for teams trying to do team science. And so you see along the bottom, there's the idea of data scientists or analysts and their role. There's the investigators, and then there's methodology researchers and maybe other collaborators of the team. And so what we're actually doing right here with the demo is we're pretending to be that middle category. So we're investigators interacting with this live running web app. Obviously, before we can do that, somebody had to write the web app. And so when I wrote the demo, I actually put on my pretend to be data scientist hat for a little bit. And so that's depicted on the left-hand side of this diagram. And I tried to author some Python source code. I used a tool building framework from the machine learning community called Streamlit. And I worked with the rest of the team, rest of my team within in source code, within the source code repo to iterate, to get at something we felt like was representative of a real interactive data analysis tool. And so once we were happy with that, we deployed it to app hosting. And that's kind of what you're seeing here, right? A live running web application. And what you'll be able to see in this demo is that from this interactive tool, you can programmatically query out clinical data from I2B2. You can pull in analysis results. In this example, we're doing pulling in results from a gene expression analysis. And then we're taking the list of differentially expressed genes and we're passing it to a gene set method called Pager, which is a method from Dr. Jake Chen's lab here at UAB. So hopefully that's good enough for an overview. And I'm not going to go into a ton of details in each of these sections, but for the first part, we just show that we can query data out of the UWS API. And we show it here just simply as a table. But I think the more interesting thing from a data science point of view or scientist point of view is that it's a data frame and you can integrate it and combine it in different ways within the tool, right? So then the next section, there was a differential gene expression analysis that was performed by another part of the team. And so we wanted to pull in the results of that analysis and basically take that list of genes that were identified there and pass it to Pager, which is then going to tell us what gene sets or networks or pathways that might be of interest to us. So I think this is where the demo gets a little more interesting, in that when we first visited this page, what had already happened was that we did all of those things. We met all those queries, and we actually already passed those results to Pager, and it's already given us back 22 gene sets that we might be interested in. And this is with default Pager parameters. But if we wanted to tune it, so if we wanted to say we're only interested in gene sets of size 85 or larger, and less than 258, right? And then you can see it dynamically requeries Pager and gives us 12 results back. And just to show, for completeness sake, we could also adjust some other Pager parameters, not all of them, just 2 representative ones. So these are just if you wanted to add or delete data sources, or if you wanted to adjust the false discovery rate cutoff. And so, so once as an investigator, you're happy with this, you might want to download it and play around with it further in another tool. So again, this is a technology demo, right? So it's not meant to be a real data analysis, but hopefully it's representative of what a team might actually do and want to do with an interactive research tool. So we're trying to set up the infrastructure with U-BRITE and put out examples to show people what might be possible in infrastructure to help with their team science.
Right, 2 minutes.
2 minutes, okay. So yeah, so for today, I just wanted to show you these 2 things, just to give an intro to U-BRITE and invite you to play around with this demo. If you're interested, it's at demo.ubrite.org. And if you want to find out more about U-BRITE, we've got a website at ubrite.org, which you're welcome to check out. We've got a lot of learning resources, training. We try to integrate with bioinformatics courses, various info courses, and others here at UAB to help people who want to use U-BRITE get started. So please don't hesitate to get in touch if you have any questions.